Okay, cool. Let's see. All right. Hi, everyone. Thanks so much for showing up to listen to me talk. And I see a lot of you here. And it's an honor to be able to be a part of this webinar series. And thanks for the team, the HETF team, for setting it up. So the title of my presentation is Steps Towards Improving Avian Disease Carrying Mosquito Surveillance in the Face of Climate Change. And I'll cover my master's work in this talk that's been monitoring at Hakalau Forest National Wildlife Refuge, as well as Laupahoehoe Forest Reserve. So I'm a master's student at the University of Hawaiʻi at Hilo in the Tropical Conservation, Biology, and Environmental Science program. And my committee members are Renee Bellinger, Catherine McClure, and Matthew Knope. So just to walk you through what I'll be talking about today, I'll start with a background on Hawaii's native forest birds and review the invasive mosquitoes and avian malaria disease system and how it interacts with climate change and other environmental factors for those of you that aren't too familiar with it. I know that a lot of you are, I'm seeing now. But I will then go into depth into mosquito surveillance and climate monitoring at Hakalau and Laupahoehoe. I'll also talk about some work we've done on innovative methods development for detecting mosquitoes, including environmental DNA and mosquito bioacoustics. And I'll finish by talking about collaborations and what we've got planned for the future. So in Hawaii, when we talk about birds, there's a story that we always tell. It's the story of the introduction of an invasive mosquito to Hawaii that over the past nearly 200 years, it and avian malaria, the disease it vectors, have been primarily responsible for devastating extinctions and population declines of Hawaii's birds. Avian malaria transmission requires the vector, the host, and the disease. Migrating birds from North America, as well as introduced non-native birds to Hawaii, likely brought the parasite that causes avian malaria. And with the introduction of the Culex mosquito, it became the link between infected and uninfected birds. So of more than 50 honeycreeper species that were present across the islands, there are 17 that remain, and more than half of those are listed as endangered or threatened species. The population declines across nearly all species have accelerated over the past 20 years, and 4 species, the Akikiki, Ake'e, Kivikiu, and Akohekohe, are on the edge of extinction, with extinction predicted within the next decade if conservation action isn't taken. So this truly is an ecological crisis. Population changes and range contractions are occurring rapidly alongside increased avian malaria infections and warming temperatures and mosquito captures across the landscape. This story is one that, like many, involves the interaction between invasive and native species that's aggravated by climate change. So historically, at low elevations, there was intense malaria transmission with minor seasonal or annual variation in infection and mosquito populations present year-round. As we move into mid-elevation forests, avian malaria was seasonally dynamic, with peak infection rates in the fall season when temperatures rose. And high elevations have been essentially disease-free refugia in forests, with only slight risk of avian malaria infection during the summer, but we've seen this changing really rapidly, and climate change is rewriting this part of the story. So since the 1950s, the mean annual temperature in the Hawaiian Islands has increased by nearly 3 degrees Fahrenheit. And as warmer temperatures reach further upslope, that habitable range for mosquitoes and avian disease reaches further into areas where resident mosquito populations haven't been before. So suitable climate conditions, along with available larval habitat, are some of the drivers that support this dynamic. There are few aquatic habitats in the world that do not provide the conditions for at least some species of mosquito to breed. Mosquitoes are flexible ecologically, able to adapt to diverse breeding sites, as well as changing climatic conditions, which has really been a key to their success as a species over evolutionary time. And even something as small as an old Spam can, a fallen leaf, or a cavity in an old tree can provide habitat for these mosquitoes to thrive. But this is also the habitat that we as humans have the most control over when managing mosquito populations. So local studies have found that larval habitat availability is a primary driver of mosquito abundance, and that forms of land use like percent of developed land, agriculture, and forest fragmentation significantly increase mosquito abundance in addition to temperature and cumulative rainfall. In wet forests, rock pools along stream beds and Hapu'u cavities created by feral pigs are the areas where mosquito larvae are frequently found. Rock pools are natural formations along stream beds, and with the fluctuation of stream flow and precipitation, these habitats are really dynamic. There's some current ongoing research led by Dennis LaPointe, also in Laupahoehoe and Hakalau, that's looking into more of how these systems can be potential corridors for invasion. Hapu'u cavities, which you see on the right, are a major component, or Hapu'us are a major component of the feral pig diet. And while we do see small cavity formation through natural processes, pigs are the primary source of excavating these larger cavities like you see here. In forests with pigs and Hapu'u tree ferns, these cavities fill with rainwater and leaf litter, leaving standing water that provide ideal breeding habitat for mosquito larvae. So together, these conditions all create a recipe for disaster. But this story of invasion and extinction is one that we're trying to change, and one that right now a lot of people, and definitely many of you that are here, are working on figuring out. So how do we change the course of this story? Options for landscape level mosquito control, such as incompatible insect technique, are being explored as an emergency response to prevent further extinctions and moving forward. But to bring these technologies from the lab and into the field, one of the things we really need is robust mosquito surveillance, and that's where my work comes in. So under current mosquito monitoring protocols in many high elevation forests, mosquito invasions could go unrecognized until it's too late. The implementation of landscape level mosquito control requires detailed population information that can only be supplied by continuous monitoring. So why study mosquitoes? Well, I think I've probably made my case for this already. In the book Mosquito, A Human History of Our Deadliest Predator, historian Timothy Weingard talks about how bashing mosquitoes has been a universal pastime since the dawn of humanity. And I think it's pretty safe to assume that we here all share a genuine hatred for mosquitoes. So just something fun that I like to I actually enjoy about it is that we all have a personal experience with mosquitoes that can connect us to them, whether we like it or not, and to the need to do something to get rid of them. The most recent common ancestor of the mosquito emerged during the Jurassic period around 182 million years ago. This period of time lent itself to the speciation of over 3,000 distinct species, and this isn't exactly the kind of diversity you may typically like to appreciate. Unfortunately, the invasive mosquito community in Hawaii is also diverse, with new species introductions of a total of 6 species occurring over the last 200 years. Recorded as a singing fly, invasive mosquito detections in Hawaii first occurred around 1826 with the accidental introduction of Culex quinquefasciatus, followed by Aedes aegypti in 1892, Aedes vexans in 1963, Waimea michellii in 1981, Aedes albopictus in 1986, and Aedes japonicus in 2003. So my study species is Culex quinquefasciatus, which is the primary vector of avian malaria. It's a nocturnal mosquito and opportunistic feeder that feeds both on birds and humans. The fact that it's a tropical mosquito means that it can't thrive in cold temperatures. And this temperature limitation is one of the primary limiting factors of its distribution. In Hawaii, the species is active and capable of reproducing year-round at low elevations and ever-increasing higher elevations. Aedes japonicus is another important species to note, and has actually been the species that I've most frequently encountered in my monitoring work. The first detection of Aedes japonicus was actually in Laupahoehoe on Hawaii Island in 2003, and it has since rapidly established throughout the islands and across elevations, facilitated greatly by their tolerance to cold, which allows them to expand their range into higher elevations. They're known commonly as the rock pool mosquito, and it can exploit streams and riparian habitat. One good thing is that it is not a vector for avian malaria, but does vector other diseases like West Nile virus. So the project that I've worked on for my thesis was funded by the Pacific Island Climate Adaptation Science Center as part of the Manager Climate Corps program. And the goal of this program is to support long-term relationships and collaborative manager-driven research to create actionable outputs for climate change adaptation. This project is a collaboration with Hakalau Forest National Wildlife Refuge, driven by answering questions that can meet the manager's most urgent needs. So my advisor, Pat Hart, scientists from around the state, managers at Hakalau, and Friends of Hakalau all work together to identify research priorities that could inform priority management actions at the refuge. So building from long-term relationships, they really set the stage for a collaboration that could continue beyond the duration of just a single student project, but really become a long-standing commitment to the refuge. And the initial project idea was to support the development and implementation of improved monitoring protocols for an early warning system of mosquito invasion. And to do this, we wanted to implement a low cost and relatively low effort protocol that could provide constant surveillance for the refuge over time. Laupahoehoe was added later as an additional site for mosquito monitoring efforts due to known mosquito and larval habitat presence, and to be able to compare across the sites to further understand mosquito invasion into high elevation forest. So some of the research questions we aimed to answer were, is there evidence of invasion of Culex quinquefasciatus at Hakalau? Do the climate conditions necessary to support local development and establishment of the mosquito and the avian malaria parasite exist at the refuge? And then how do they vary across the elevational gradient? How are Hapu'u cavities distributed across the refuge? And if there are trends in mosquito larval occupancy in these cavities and the factors that might influence that. And finally, looking at innovative methods, monitoring methods, and if they can improve detection efficiency. So the lines of action that we followed were conducting adult mosquito monitoring, climate monitoring, and also larval habitat surveys. So past mosquito monitoring at the refuge was published in 2016 by the USGS and HCSU, led by Dennis LaPointe, and this provided a comparison of avian malaria prevalence and mosquito occurrence from 1998 to 2012. This study found limited local transmission at high elevations, general increasing prevalence at lower elevations, but also a twofold decrease in prevalence of avian malaria from 1998 to 2012, attributed to a variety of different factors, one being a colder climate decade and also reduction in larval mosquito habitat. During their work there, they did not have any detections of larval Culex, and only one adult capture at 1,313 meters. So the results showed a limited presence of Culex at the refuge, but they did document the establishment of Aedes japonicus in both Hapu'u tree fern cavities and rock pools in the lower reaches of the refuge. And they finished their report with the question of wondering why hasn't Culex become established at Hakalau, which this is really something that a study that our work tried to follow up on. So as many of us know, Hakalau is a stronghold for native Hawaiian forest birds and historically has had positive or stable long-term trends. And a report was just released this year that analyzed population trends across the refuge from 1987 to 2019. across open forest, closed forest, and pasture. And while some forest bird species continue to show upward or stable trends, findings show over that entire time period, findings showed that since 2012, most species populations are declining across the forest strata, and particularly populations at lower elevations, showing that the conditions at the refuge are likely changing. So at Hakalau, we established 2 transects in the Puakala and Hakalau tracts of the refuge, extending from 1,600 to down to 1,300 meters in elevation. Existing bird survey transects established in the 1980s, I believe, and visualized here as the black dots extend to the lower fence line of the refuge, which are the black lines. And our 2 transects extend an additional 3 kilometers below the lower fence line. The pink dots here represent the mosquito traps that we have along those transects. So this elevational gradient represents a transitional zone from mid to high elevation forest where you may expect to first detect signs of mosquito invasion. And we're especially interested in this area to be able to monitor some of the lower reaches of the refuge that are a little bit harder to get to. At Laupahoehoe, due to easier access to lower elevation sites, we were able to extend the transect lower from going from 1,100 up to 1,500 meters. And this transect was set 50 meters off of Blair Road. So you can see the purple triangles there in reference to where the transect is located to Blair Road. And due to the lower elevations, closer proximity to forestry roads and urban development than Hakalau, we expected to have higher capture rates and see more evidence of invasion at this site. So for a summary of our methods, we set out a total of 51 passive gravid 80s traps across 300-meter elevational gradient. 3 climate sensors were placed on each transect for a total of 9. And Hapu'u tree fern cavity surveys were conducted along belt transects. For adult mosquito monitoring, we monitored for evidence of invasion using these passive traps, validated the capture rates of the passive traps at low elevation, high mosquito density sites, and conducted comparative trap efficacy experiments between active and passive traps. Mosquito trapping isn't quite as straightforward as you might think. Mosquito traps are designed to exploit the ecology of a target mosquito to lure it into the trap using chemosensory and visual cues. So there are several trap types, and these traps are divided into 2 categories. The traps we use here, known as gravid traps and host-seeking traps. So gravid traps use a combination of olfactory and visual stimuli to lure a gravid female mosquito seeking a place to lay her eggs, while host-seeking traps, like the one on the right, utilize CO2 to simulate a living host and lure a female mosquito that's seeking a blood meal. So these traps can also be divided into active and passive distinctions. The 2 main active traps cited in the literature and used across Hawaii include the CDC gravid trap and the BG Sentinel-2. These traps are typically paired with sites, with the BG considered to be the gold standard, with a fan in addition to other lures that actively pull the mosquito into the trap. Some of the disadvantages of these traps are really heavy. They require a power source and they have to be operated by a field crew daily. So just try to imagine carrying, or maybe many of you have carried this equipment on your back into the forest, and you'll notice that not pictured here are the 12-volt car batteries that they run on, or 6 volts sometimes. So due to field constraints, we chose to use the passive gravid 80s trap, and these traps are really simple, low cost and lightweight. They don't need power to run, and they can be left out for long periods of time, which makes them really ideal for trapping at sites like Hakalau. The mosquitoes, a lot of the same cues are used here. So mosquitoes are attracted to the black color of the trap, as well as the odor from the stinky water infusion. And once they fly in, they struggle to get back out, eventually getting stuck to a dangling sticky card within the trap. But one major disadvantage is that they are considered passive because they rely on the mosquito to trap itself, which may have costs in trap efficacy. And this has been seen in other sites, although it hasn't been validated or tested much locally in Hawaii. So for a low elevation trap validation, we went to the UH campus and UH Hilo farm to test the efficacy for these traps in capturing Culex mosquitoes. Oops, sorry about that. And traps were shown to be effective in capturing Culex, although there is significant bycatch on sticky cards. So sometimes you'll just get a card loaded with calophorid flies, which makes it really hard to try and find mosquitoes and can also reduce some of the trapping power. But these results contextualized and validated the functionality of the traps on one level for us to be able to implement in high elevations. So in 4,971 trap nights over 2 seasons at Hakalau and all of the traps that we had 0 mosquito captures. So Aedes japonicus were observed. They were the only mosquito that were observed in the area, and they were seen on the landscape, but not actually found in the traps. And these observations were also made by Dennis Lapointe during trapping at Hakalau in 2016. And Aedes japonicus is also known for being difficult to trap in conventional traps. At Laupahoehoe in 1,409 trap nights, we captured 1 Culex and 21 Aedes japonicus. And all of these mosquito captures were made below 1,335 meters of elevation, which is the portion of the transect that actually extends below where we've been able to trap at Hakalau. So while these numbers are really low, it's a good indication that mosquitoes are still at low densities at these sites, but we also have to consider trap efficacy when looking at these results and the possibility that these passive traps that we were using were not actually detecting mosquitoes present in the landscape. So to follow up on that question, in November of last year, we conducted trap efficacy experiments. So pairing the active and passive traps side by side, as well as a GAT trap to look at, to compare performance. And during these experiments at Hakalau, we captured 1 Culex mosquito at 1,671 meters in an active BG trap. One other detail too is that these at Laupahoehoe and Hakalau, they weren't run at the same elevations just due to accessibility. So at Hakalau, the transect was actually from 1,600 to 1,900 meters that we were running them. While at Laupahoehoe, it was from 1,100 to 1,400. But this, to our knowledge, is the first capture of a Culex mosquito in this portion of the refuge. And some research supports that there's a possibility of high elevation mosquito captures like this to be windblown, but instead of an indication that resident populations are established, but it's really difficult to tell with something like a single capture, and just emphasizes how important it is to have continuous monitoring in these areas. At Laupahoehoe, we had 6 Culex captures in BG traps below 1,200 meters, and 6 Aedes japonicus trapped across trap types. So for climate monitoring, we wanted to assess the daily, monthly temperature conditions needed to support local Culex, Quincke fasciatus, and Plasmodium relictum development across the elevational gradient. Hobo data loggers logged temperature and relative humidity every 10 minutes since February of last year. And this is the data from the highest elevation monitor that we have at 1,510 meters in Puakala. Culex mosquitoes require 9.9 degrees Celsius for development, and Plasmodium relictum requires 12.97 degrees Celsius, which I've included these temperatures as the dotted lines to represent temperature thresholds for development. And if we pay attention to the average temperature line for each elevation and where it passes above the threshold, these are the months that provide the conditions necessary for vector and parasite development. So based solely on these temperature thresholds, climate conditions revealed that mean ambient temperatures necessary for culex are present year-round, with conditions for Plasmodium present 5 months out of the year. But it's also important to note that this is minimum temperatures below some of these thresholds may still limit development, and the refuge is still considered to be a low-risk transmission zone. So for Laupahoehoe, I included the graphs for temperatures across the elevational gradient. And here we can see the trends that also generally apply to Hakalau's annual temperature regime. And some of the takeaways are that there's a 0.5 degrees Celsius temperature increase for every 100 meters and a 1 degree Celsius increase across the elevational gradient. And there have been many models that have predicted these conditions as well. And so these climate sensors are giving us localized data that's really affirming and confirming some of the things that we already knew. So for larval habitat surveys at Hakalau, we mapped the distribution and density within fenced and unfenced areas to provide a Hapu'u distribution map to the Hakalau management to aid in management decision-making and mosquito larval habitat reduction. And at both sites, we characterize the cavity characteristics, so water holding capacity and persistence and patterns of larval occupancy over time. The tree fern cavity surveys were done on belt transects, and measurements taken for each Hapu'u cavity included length, width, and depth. They were characterized as holding water or dry, as well as given a subjective age grade. And additionally, if the Hapu'u cavity was holding water, it was inspected for larvae, and if the larvae were present, they were collected and reared to adulthood for species identification. The subjective age grading is based on standard operating procedures and includes fresh, intermediate, and old classifications for cavities that vary in the amount of starch and level of decay, the turbidity of the water, and presence of leaf litter, which are all factors that may contribute to whether a cavity is occupied or not. So the Hapu'u cavity surveys at Hakalau covered 10.5 hectares across both fenced and unfenced tracts of the refuge, and revealed extremely low densities of Hapu'u cavities, with 89 cavities total identified and a density of 8 cavities per hectare. Given the survey area, disproportionate 73% of the cavities were found in unfenced areas, despite representing only 33% of the total area surveyed. 27% of the cavities were holding water, and only 1 cavity was occupied with Aedes japonicus larvae. The surveys that were conducted within the fenced areas were a snapshot survey, whereas the cavities that were located along our transects, we were able to monitor over time. And no culex larval detections were made, and also no Hapu'u cavities were found in the fenced section of Pu'akala. So all of the surveys were conducted along these yellow lines across the refuge. In an initial survey at Laupahoehoe of 0.425 hectares, we found 74 Hapu'u cavities, and this is a density of 176 cavities per hectare, so much higher than Hakalau. Since the initial survey, we've marked 35 Hapu'u of all age classes that hold water and have visited them each month to track patterns of larval occupancy. So far, we've found that cavities age rapidly, changing from fresh to old in the matter of months, and seem to remain stable at old age. The fresh cavities are actually really hard to come across, and to determine the age or the persistence of these older cavities over time, I think we'll need a lot longer study. So this is hopefully something that we can do at Hakalau with Hapu'u cavities that have been marked going back in a few years' time and seeing which ones are still actively holding water or able to support larval habitat. All larvae collected so far have been Aedes japonicus, and we've seen some seasonality and variation in occupancy as you would expect. And in July, for example, 35 of the 35 cavities, only 5 that had water were not occupied by Aedes larvae. And so far, no Culex larvae have been detected. So the time in the field has allowed us to learn a lot. We've observed changing climate conditions, moving into higher elevations that can support both Culex development and avian malaria transmission. We found evidence that Hapu'u cavity densities are low at Hakalau, especially in fenced regions, and Hapu'u cavities don't really appear to be a significant contributing factor to available larval habitat within the areas that we surveyed as compared to Laupahoehoe. We've seen the evidence of Aedes japonicus invasion, which supports past observations and the possibility that Aedes japonicus could serve as a proxy indicator for Culex invasion in a warming future. For adult mosquito trapping, we learned a lot about the challenges of trapping in remote, low mosquito density sites and the disadvantages that may come with using passive traps, as well as the need to understand how traps perform in relation to each other. So our mosquito trap numbers are so low that we can't make many conclusions about invasion, but we're combining our data with data collected from around the state to compare among trap types. And if we can understand exactly or more about how GAT traps perform compared to active traps, we can hopefully increase their utility to contextualize captures and still get the benefit of using these traps in a future where mosquito monitoring needs are only increasing. I think they would fill a gap that is really needed in terms of reaching sites and being able to have field teams on site checking traps. Other things that we can do are to look at ways for improving passive trap efficacy, as these technologies are always evolving and with the more that we know. But for now at Hakalau, this fall we'll be deploying a long-term mosquito monitoring design that's supported by the Friends of Hakalau and our learnings from this project, using 6 permanent BG CO2 traps that will be run year-round. So there's some new developments in CO2 timers and solar batteries that are really going to allow us to use these higher efficacy traps over a long period of time. Our project has evolved a lot over time, and many new ideas have sprouted from the challenges that we encountered in these efforts. And some of these results have allowed us to apply what we've learned to try and improve the efficacy of monitoring through innovative methods development. One project that I've worked on that was ongoing at USGS is it's a collaboration with the USGS Pacific Islands Ecosystem Research Center that uses genetic tools, lab experiments, and field samples to develop, optimize, and validate a species-specific eDNA assay that can target a gene for the detection of Culex in aquatic larval habitat. Environmental DNA is a non-invasive surveillance tool that's capable of detecting trace DNA of target species in water, soil, and air, and it can be acquired, samples can be acquired by taking a water sample from a site of interest, like a Hapu'u cavity, filtering the sample, preserving it, performing a DNA extraction, and finally running a quantitative PCR assay to determine presence or absence of the target species within the sample. The application of eDNA is particularly relevant in high elevations where mosquito detection is challenging due to seasonal and low density populations, like I've talked a lot about already. And in these scenarios, larval habitats that may appear to be unoccupied, but they actually still have remnant DNA from recently emerged mosquitoes that have left larval or pupal casings behind. So our questions are around if the eDNA assay is specific enough to Culex, trying to understand the biotic variables that influence detection, and defining the parameters of detection as well. The methods that we're using to develop the assay include designing a species-specific primer probe for the CO1, cytochrome oxidase 1 target region, and doing this also for Aedes japonicus. The in silico analysis requires alignment of this target region to test for the specificity of the assay to just detect the species that we want. Oh, sorry about that. And development of sampling protocol and assay parameters in the lab to finally be able to take the technology from the lab and into the field to test and validate for detection. Finally, as the LOHE lab is a bioacoustics lab, it only made sense to try and get our hands on some mosquito recordings. So not nearly as fun to listen to as the bird recordings, but maybe the only time that a mosquito buzzing in your ear might be a good thing. This summer with a PIPES intern, Olivia Boucher, we began testing the feasibility of using these small acoustic recording devices called audio moths, like in this picture, to detect mosquitoes as a way to increase efficacy of passive trapping. So we used the QLEX lab colony at University of Hawaiʻi and wild-caught Aedes japonicus mosquitoes to create an acoustic sound, or begin to create an acoustic sound reference library for both female and males of each species. And then we used Raven Pro bioacoustic software to make selections of species-specific wing beat frequencies. Our lab recordings were successful, and that we were able to find differences between both species and sexes. However, unlike birdsong, they're pretty difficult to recognize by the human eye and may need machine learning to accurately detect and process the data. So we're just in the beginning stages of playing around with this, but this is an example of the female QLEX. And then here we have an example of the male. Oh no, now I'm on a loop. Okay. So from the lab, we went to Hilo farm and launched gat traps modified with audio moths, and check the sticky cards on the traps every day along with the recordings to confer captures. So we're still going through a lot of these recordings a single night because they run from 4 to 6 a.m. Or provide 14 hours of recordings, which is a lot. But we do have some observations that show using audio moths in urban environments, which was one tricky piece here, is complicated because of ambient noise that can overlap with frequencies where we would see the mosquito wing beat, like other loud insects, flies, or just in the area that have actually been caught on the sticky card, as well as crickets, coqui frogs. So you can listen to an example here. So in these recordings, you can see that in the area of the spectrogram that we would expect to see the mosquitoes appearing, it's actually obscured by some type of ambient sound. And rain could also be a challenge here. So you can see it's not quite as clean to be able to detect something as small as a mosquito. But in places like Hakalau that are virtually silent at night, we actually may be able to make these detections more easily, which suggests that if the recorders were optimized, that audio moths could augment the efficacy of these passive traps. Here's the sound of Hakalau at night. Pretty darn quiet. So there's a lot of potential for acoustics to be used in the future. And I know that a lot of work is being done with this currently with sound lures for gat traps or for trapping as well. So I'm really excited and grateful that this work will be continued by an incoming graduate student with the support of PICAS and Friends of Hakalau too. So this project has brought together the university, the refuge, the community, and the state in a collaborative partnership, and also relationships with complementary projects, including another PICAS-funded project with USGS Pierce, the Kauai and Maui Forest Bird Recovery Projects, and Big Island DOFA team. They've all facilitated so much of this work. And an extra special thanks to PICAS and Friends of Hakalau for supporting it, and also to Dennis LaPointe, who's given so much expertise, generosity, and guidance along the way. When I look around at the community of people that are working to ensure a healthy future for Hawaiian forest birds, I'm really blown away by the passion and commitment. And we've got just about every type of person on the team, from geneticists in the lab to bioacousticians and modelers and field workers on the ground. So I think we've got a pretty solid team. And thanks so much for listening. I'm happy to take any questions if we have time.
Great. Thank you so much, Stephanie. I don't see any question. Wait,